Welcome to Principles of Ecology. Today, we are going to explore one of the most powerful tools in population ecology, life and fecundity tables. Imagine trying to predict whether an endangered species will survive or if a pest population will explode. How do biologists actually do that? They do not just guess. They use precise mathematical bookkeeping. By the end of this video, you will understand exactly how to track the survival and reproductive success of any species, from birth to death. Let us dive in. To understand population dynamics, we first need to understand what a cohort is. Think of a cohort as a specific generation, like a group of 100 newborn rabbits, all born in the same forest at the same time. A life table is essentially a detailed diary of this cohort. It records exactly how many individuals are alive at the start of each age stage and how many die before reaching the next. This systematic tracking is crucial because it reveals the specific survival and mortality rates at different stages of life, giving us a clear picture of the population's overall health. Now that we know what a life table is, why is it so important? Understanding these tables is absolutely critical for real-world applications, like wildlife conservation and pest management. For an endangered species, like the giant panda, life tables pinpoint exactly which age group faces the highest mortality, telling conservationists exactly when the animals are most vulnerable. Conversely, if we are dealing with an invasive crop-destroying insect, these tables reveal the best time to apply control measures. Even human demographers use these exact same principles to plan for healthcare and infrastructure based on aging populations. This brings us to the mathematical representation of life tables. The most fundamental metric we calculate is survivorship, denoted by LX. Conceptually, this is just the fraction of the original population that survives to a given age. In the formula, NX represents the raw number of individuals still alive at age X, and N0 is our starting cohort size at time 0. By dividing the current survivors by the starting number of babies, we get a proportion. If LX is close to 1, almost everyone survived. As age increases, this value must always decrease because individuals can only die, not come back to life. While survivorship tracks who lives, we also need to measure who dies. This helps us pinpoint when a population experiences the most stress. We use two formulas here. First, dx represents the absolute number of individuals that die during an age interval. We find this simply by subtracting the number alive at the next age, nx plus 1, from the number alive at the current age, nx. Next, we calculate qx, the age-specific mortality rate. By dividing the number of deaths, dx, by the number of individuals who started that interval, nx, we find the probability of dying during that specific time. A high qx indicates a very dangerous life stage. Before we move on, let us see this with a concrete example. Imagine a researcher tracking a cohort of 500 newborn field mice. This is our N0. By their first birthday, at age 1, only 100 mice are still alive. This is our N1. We want to find the survivorship to age 1, or L1. We use our formula, dividing N1 by N0. Plugging in the numbers, we divide 100 by 500, which gives us 0.20. Physically, this means a newborn field mouse has only a 20% chance of surviving to its first birthday, highlighting how difficult the first year of life is in the wild. Now that we have the survivorship, let us calculate the mortality metrics for these same mice during their first year. First, we need the absolute number of deaths, D0. We subtract the 100 surviving mice from the original 500, revealing that a staggering 400 mice died. Next, we find the infant mortality rate, Q0, by dividing those 400 deaths by the starting population of 500. This gives us 0.80. This means there is an 80% probability of dying during the first year of life. This massive drop is very common in small mammals that produce many offspring but provide little parental care. We have thoroughly examined survival and death. But what about new life? While a life table tracks death, a fecundity table tracks birth. Here, we introduce a new variable, mx, which represents the age-specific fecundity. This is the average number of female offspring born to a single female during a specific age interval. However, there is a catch. A female can only give birth if she actually survives to that age. Therefore, we must multiply her probability of surviving, Lx, by the number of offspring she would have, Mx. This product gives us the realized fecundity, a crucial concept for understanding true reproductive output. This concept of realized fecundity leads us to the most important number in population ecology, the net reproductive rate, denoted by R0. Conceptually, this formula calculates the average total number of female offspring produced by a single female over her entire lifetime. The Greek letter sigma tells us to take the sum of the realized fecundity, Lx times Mx, for all age classes. We're multiplying the chance of surviving to an age by the birth rate at that age and adding it up for the whole lifespan. If R0 equals 1, the population is perfectly stable. If it is greater than 1, the population is growing, and if it is less than 1, it is shrinking toward extinction. Let us apply this powerful formula to a real-world scenario. Imagine we are studying a small population of rare birds to see if they are recovering from endangerment. We have data for 3 age classes, 0, 1, and 2. The survivorship values are 1.0, 0.5, and 0.1. The fecundity rates are 0 for babies, 2 for age 1, and 4 for age 2. To find R0, we set up our summation for the entire lifespan. We will multiply the survivorship at each age by the birth rate at that same age, and then add the results together for all 3 years. Now, let us execute the calculation we just set up. We plug in our specific numbers. For age 0, the birds are alive but have 0 babies, so 1.0 times 0 is 0. For age 1, half the birds are alive and have an average of 2 babies, giving us 1.0. For age 2, only 10% survive, but they have an average of 4 babies, giving us 0.4. Adding these together, 0 plus 1.0 plus 0.4 gives a final R0 of 1.4. Because 1.4 is strictly greater than 1, we can confidently conclude that this endangered bird population is successfully growing and recovering. To visualize everything we have calculated, ecologists use survivorship curves, like the ones shown in this diagram. Let us walk through what we are seeing. The horizontal axis represents age, and the vertical axis represents survivorship on a logarithmic scale. Notice the 3 distinct curves. The top curve, type 1, represents species like humans, where most individuals survive to old age before mortality sharply increases. The middle straight line, type 2, represents species like birds, which have a constant probability of dying at any age. Finally, the bottom curve, type 3, drops steeply right at the start. This represents species like our field mice or frogs, which produce many offspring but experience massive infant mortality. We have covered a lot of ground today, so let us summarize the most important ideas. First, life tables are the ultimate bookkeeping tools of ecology, allowing us to track the survival and mortality of a specific cohort from birth to death. Second, by adding fecundity data, we can calculate the net reproductive rate, R0, which tells us definitively if a population is growing, shrinking, or stable. Finally, remember that these are not just abstract numbers. Transforming raw field observations into mathematical predictions provides the essential data needed to save endangered species and manage ecosystems effectively. You now have the foundational tools to understand population dynamics.